All right, hi everybody. For the purpose for the week 7 work is largely to talk about the innate immune system, um, which we did briefly talk about last week, and we actually mentioned a lot of the components embedded within the innate immunity process. But now we're going to kind of put them together into one package. And then just to remind you that there is like another line of defense in addition to the innate immunity and the adaptive immunity is that we have our intrinsic first line of defense, which is our skin and our mucous membranes. So we have, for example, our epidermis, which is a physical barrier. The cells at the top of that barrier are dead and basically just a bunch of protective keratin. Even if there was some material that the bacteria could live off of those cells, the bacteria would fall off the body because those cells are shed regularly. And then in addition to those dead cells at the top, we have the sebum and the sweat, these chemicals that are there on the surface of the skin, in which the sweat is creating this very salty hyperosmotic environment. The sebum is this very low pH material that will obviously also impair the growth and reproduction of any microorganism. Our mucous membranes, which are basically every other epithelium in our body. They're usually lining some sort of tube in our body. They are generally producing mucus, and mucus is this viscous material that will trap microorganisms and debris within it. This is most dramatic when you talk about the mucociliary escalator in the respiratory system. So, for example, in the trachea, if you remember your trachea from last term, there's that epithelium lining, obviously, the tube of the trachea, but then they have these little cilia that are off of the surface projecting into the lumen of the tube, and all of those cilia are beating in this uniform fashion, and so all this mucus that's in the tube, and of course any debris that happens to be trapped within that mucus, is going to be crowd surfed up towards the top of the trachea, where eventually they'll end up in the digestive system and then go out of the body in that regard. So you can move the mucus, which traps the bacteria, and then sweep them away with the cilia into the digestive system so that they can be removed from the body. We've also, in this course, talked about the lacrimal apparatus, so the production of tears in the superior lateral region above the eyes. We know that those tears contain an antibiotic called lysozyme present in it. This is an antibacterial agent that targets bacterial cell walls and breaks them down. And then we're constantly, as you recall, blinking to wipe the tears or wash the tears across our conjunctiva and protect that very vulnerable epithelium from infection. Saliva has various antibacterial chemicals within it as well. Urine is actually, as you've covered in this class, shouldn't be having much in the way of bacteria in it, and in fact, the flow of urine through your urinary system will cleanse the urethra and inhibit microbial growth in the urinary system. Urine is in most cases, antibiotic. Defecation and vomiting, that's pretty obvious. The simply expelling material from your digestive system in a mass sense will remove any microorganisms that are present within there. And then even more specifically within the digestive tract, the gastric juices of the stomach have this incredibly acidic pH and also contain all these digestive enzymes, which would be basically making the stomach a very inhospitable place for germs. So that's our intrinsic line of defense. Let's move on to the innate side of the defense. This is our second line of defense. These are the first example of which I'll use is the interferon pathway or the IFN pathway. I talked about this last week, where you basically can imagine an epithelium. So we have like a already tissue, but epithelia is obviously the easiest to draw. And we have, say, 4 cells in that epithelium. And unfortunately, some pathogen, let's say a coronavirus even, is attaching to the surface of that epithelium, and it injects its DNA in, and now we've got a cell that is infected with the coronavirus. And so this cell actually has a pathway in it to recognize that it has been infected. And once it quote unquote knows that it is being infected, it's going to start releasing these basically signaling molecules called interferons into the interstitial space. All the cells in this tissue are going to have interferon receptors. So these are the interferon receptors. And as a result of these interferons released from the infected cell binding to the interferon receptor of a neighboring or nearby cell, that cell will start producing antiviral proteins, or AVPs. I'll write that down here as well, antiviral proteins. And those AVPs are going to make it harder for that other coronavirus that's probably also in the neighborhood to be able to successfully initiate some sort of reproduction, because these antiviral proteins are going to inhibit some stage of the viral pathway. So this cell in the process is going to die. It kind of knew that already because it was infected, but before it did so, it released all these warning chemicals, the interferons, to tell the neighbors, hey, you need to start producing antiviral proteins because a virus infection is coming for you. Another major part of our second line, or our innate defenses, is called the complement system. We also talked about this last week, although we'll go over it in a little bit more detail here. The complement system is a defensive system that consists of about 30 proteins. All of these proteins are made in the liver, and they're all constantly circulating through your bloodstream. So at any moment you would have these complement proteins swimming in your blood, and they're just waiting to become activated. As you can see here, this is a reasonably complicated pathway. So we have this inactive blood protein C3, which under certain circumstances will break up into C3A and C3B, which will then combine with C5. C5A breaks into that, and then you need a C6. So as I'm reading this, I hope most of you were just rolling your eyes and thinking, oh my God, you don't expect me to know this. And of course I don't. I want you to know that there is this series of inactive blood proteins, these complement proteins that are just constantly swimming around your body, and then through a number of complicated pathways, they become active. And the result of complement becoming active is 3 possible outcomes. One is called, as you know, phagocytosis. So the complement system will start tagging proteins onto the microorganism that it comes into contact with, and those tag proteins that complement added will make this microorganism become very attractive to a phagocytic cell, which will then digest the microorganism. This addition of proteins to a microorganism to enhance the phagocytosis process is referred to as opsonization. And we'll talk about that a lot next week when we start talking about antibodies. So, antibodies also carry out opsonization. The second thing that complement does is inflammation. And I won't spend too much time talking about that here because the next lecture will be on inflammation in detail. But basically inflammation will, as the slide says, increase blood vessel permeability, increase actually the attraction of a region of the body to various phagocytic cells, the neutrophils in particular. So that's what complement does, phagocytosis, inflammation. And the third one is actually the coolest, which is called cytolysis, where basically a combination of these complement proteins insert themselves into the membrane of the microorganism, essentially creating this channel which will break down all those sodium and potassium and proton gradients that are supposed to exist across the bacterial membrane, and as a result of that, the bacterium will die. So those are the 3 things: opsonization and stimulation of phagocytosis, enhancing the inflammatory process, or in fact initiating it, and then also cytolysis, so directly killing the microorganism. Now, what actually gets the complement pathway activated? Well, there's actually 3 different components to this, or 3 different ways in which it can become activated. The first is called the classical pathway, and this occurs when an antibody binds to an antigen. Now, these are not terms we have talked about very much in this class. The antibody-antigen complex is what we'll be doing basically the entirety of our work with next week. But basically, an antibody is a protein that you have made that will bind to a specific antigen in the world, and the antigen is usually in a very general sense, the pathogen itself. So the antibody is kind of tagging the microorganism. And just this simple interaction of the antibody tagging the organism will trigger the activation of complement. So we get this formation of these antibody-antigen complexes, which will create the classical pathway to turn on the complement. The second way to turn on complement is the alternative pathway, and this turns on complement via microbe-specific lipid carbohydrate complexes on the surface. So bacteria and various other microbes will have lipid carbohydrate complexes that are unique to those microorganisms. And a better way of saying it is they don't exist in humans. And as a result of that, again, it makes a good target for turning on complement. And so complement will be turned on that way as well. And then complement can also be activated by a molecule called lectin. Lectin is produced by the liver when a macrophage releases certain chemicals after they've phagocytosed the newly discovered invader. So basically, macrophages do their job, they phagocytose something, they release this chemical when they actually do phagocytosis, and that chemical will then reach the liver, and the liver will then produce lectin. Lectin, once entering into the bloodstream, will then also initiate the complement pathway. So you could be activated by, in the classical pathway, the adaptive response through its antibodies, through the alternative pathway by the actual bacterium or the pathogen, or in the lectin pathway by our macrophages, which is really the heart of our innate response. We have a couple other smaller lines of defense, but I still want to mention them. The first group is called iron-binding proteins. Basically, the idea is if you have free iron swimming around in your body, that would actually be very useful for bacteria. Bacteria desperately need iron to live. And so, as basically in regions of your body where you store or transport iron, you need to be able to sequester it. Another way to say that is you need to find a way of protecting it so it won't be available to any bacteria that or other microorganisms that are in your body. There's a few examples of these iron-binding proteins listed here. Transferrin and ferritin are involved in the storage of iron in plasma, interstitial fluid, and the liver. And if you recall, we actually talked about this in Biology 1160 when we talked about destroying old red blood cells and then trying to recycle the iron from the heme groups. So you'd have to, when you're transferring iron from the site of red blood cell destruction to the liver, you need to sequester the iron by binding it to transferrin. And then once it's stored in the liver or in the red bone marrow, for example, the iron will then bind to ferritin, which will then, again, sequester it. Lactoferrin is doing the same thing, except in milk, saliva, and mucus. Hemoglobin, you know what that's doing. Of course, that's sequestering them in red blood cells. And myoglobin is doing the same thing to sequester oxygen and iron in muscle cells. We also have, just as a sort of aside here, this fourth example of an innate defense, which is the antimicrobial proteins, or AMPs. This is a small, these are a pretty large group of proteins that are very small, but they are multifunctional. Dermacidins are produced by the sweat glands, and actually this is what they look like. They create, again, a channel in the membrane of the bacterium which will break down all these concentration gradients, and the bacterium will die. Defensins are naturally released by our neutrophils, macrophages, and actually just regular epithelial cells. Cathelicidins are released by neutrophils, macrophages, and epithelia as well. And finally, thrombocidin is released from platelets. Of these, the dermacidins and the defensins, I would say, are the ones that will most commonly be identified. In terms of what they are actually doing, I would say that there are really two functions to these antimicrobial proteins. One, which is obvious, is that they should kill the microorganism in question. And two, is that they need to attract dendritic cells and mast cells to the to the scene of the crime. The dendritic cells are also phagocytes, and the mast cells, as we talked about last week, are responsible for releasing histamine. And histamine is all about the inflammation story that we'll be talking about in the next lecture. I do want to mention the natural killer cells. These are the smallest and least known of the lymphocytes. So we'll talk about its cousins, the T cell and the B cell, I should say siblings, next week. These natural killer cells are found in the spleen, in the red bone marrow, in the lymph nodes, and of course, obviously, through the blood. The natural killer cells, as you can see in the schematic here, but actually probably way more exciting in like the real picture, the electron micrograph that's being artificially colored here. The NK cells actually directly attach through receptor interactions to some foreign cell, and then as a result of that interaction, will then start releasing these antimicrobial chemicals. The two chemicals that they release are called perforin, which again puts a hole in the membrane for the same sort of effects that you saw with those other proteins earlier, and also releases an enzyme called granzyme. And granzyme will cause the target cell to undergo apoptosis. And apoptosis is basically a mechanism through which cells can kill themselves. And so as a result of these two proteins, the perforin and the granzymes, of course, the intention is to kill the target cell that the NK cell has been attached to. The other cells that we need to consider in the innate defense are the neutrophils and the macrophages. We've covered these last week. These are the cells that will be right at the point of injury when it happens, the first responders at the scene of the crime. And remember, the key word when you talk about neutrophils and macrophages is that they are the two big phagocyte cells that are going to arrive to the scene and basically start clearing up the debris and hopefully capture some of the microorganisms that are present there as well. So basically, the moment you're injured or your body thinks it's infected, neutrophils and monocytes will go to that location directly. The monocytes will then become one of two things. They can either become, so these are the macrophages, which are derived from monocytes, as you know, and they can become wandering or fixed macrophages. Wandering macrophages are the ones that are actively undergoing phagocytosis, so they are digesting stuff. Whereas the fixed macrophages are more sentry-type macrophages. They will stand in defense of currently uninvolved tissues. You've seen many examples. These are the Roombas that I'm talking about all the time that are crawling along the surface of the various tissues in your body. So, for example, the Kupffer cells in the liver that we talked about at the start of the course, in 1160, and the microglial cells in the central nervous system, which we did cover in this course in our first week on the nervous system. But really, when you're talking about the neutrophils and the macrophages, the story is one of phagocytosis. And we talked about this at length last week. Again, the first stage of phagocytosis is chemotaxis. So if you have a nasty microorganism over here, and you have your phagocytic cell over here, how does the phagocytic cell know to go in this direction and not into this direction where we have, one of our nice happy cells? Or maybe it's an epithelium of happy cells. How does this phagocyte know to go in this direction and not into this direction? And the reason is that by hook or by crook, either by its own regular activity or through the actions of other cells in your immune system, chemicals. And the phagocyte is able to detect those chemicals and kind of follow it. Like, if you smell something really good, you're able to walk in the direction in which you're able to smell that thing. That process of walking in the direction of a chemical concentration gradient is what chemotaxis is. Once you have chemotaxis, the microbe will then adhere to the surface, or rather the phagocytic cell will adhere to the surface of the microorganism, where it will then ingest it and bring it into the cell as part of a phagosome. The phagosome will then fuse with a lysosome of the cell, which will destroy the microorganism, leaving you with this residual body which contains the debris and the indigestible materials of the microorganism. And then as a final stage, this will become exocytosed, and the residual body will release all the debris into the extracellular milieu. Okay, so that's the end of our brief overview of innate immunity. In the next lecture, as I've been promising, I'll talk about the inflammatory process, and I'll see you then.